I researched microplastics on Nantucket Island. We wanted to do a baseline study, and so we quantified the presence of microplastic pollution through looking at seawater, intertidal beach sediment, and shellfish. So to do that, we chose 3 different locations on the island, specifically Nantucket Harbor Beach. There's a lot of activity going there. You have commercial fisheries, you have commercial boating, there's also recreational boating, and there's houses, residential properties, and commercial properties lined up against that area. The second location was Nantucket Beach, which is over here. There we collected seawater, beach sediment, and also bay scallops in November of 2019. This place has less activity than Nantucket Harbor. It is a little bit enclosed, similar to here. There is a bay scallop fishery here, and there are boat moorings as well. The third site we did was Surfside Beach, which is very opposite from these two on the other side of the island, open to the Atlantic. Here, mostly people just do swim, they walk, and they surf as well. There's also a seal population. So we collected water and sand samples from all 3 locations, oysters from Nantucket Harbor, and bay scallops from Nantucket Beach. We brought these samples back into the lab. With the water, we filtered it through 20 micron Nitex mesh, and then we looked at those, we filtered it, and then we put those contents onto a glass slide, and we looked at that under a microscope. For the sand, we collected it along a 100-meter transect at 10 sample points, and we collected the top 2 centimeters, and then we brought that back to the lab, and we sieved it through a 4,000, 2,000, and 1,000 micron sieve set. Everything that went through the 1,000, so that was less than 1,000 microns, was then processed using density separation. So you take a subsample of sand and you mix it with a concentrated solution of sodium chloride. And most plastics are less dense than salt water and sodium chloride, so they will rise, and you can decant that off onto a filter, and then you can view that under a microscope. And then the shellfish we collected along that 100-meter transect where the sand was collected. They were picked at random, brought back to the lab. They were digested with the enzyme trypsin to break down the organic matter. And specifically, we looked at the oyster, including the pseudofeces, just the oyster digestive tract, and the bay scallop. And so what we found was, for the seawater, all samples from all 4 sampling dates, which included June 2019, November 2019, May 2020, and September 2020, all contained some level of microplastic pollution, as well as the sand samples. All sample dates and all sites contained some sort of level of microplastic pollution. And this is showing data for below 1,000 microns and also above 1,000 microns up to 4,000 microns. We also found that there were some significant differences in microplastic concentrations in the seawater between 2019 and 2020. And the reason why we separated these years is because COVID-19 pandemic happened in 2020, and we noticed that some of our counts for these May 2020 and September 2020 months were lower, so we wanted to compare the years. And what we found was that overall in 2019, for both the seawater and sediment, microplastic concentrations were higher. And in 2020, for seawater, the concentrations in 2020 were significantly lower. And that may be due to less people being on the beach, less people being on the island, hypothetically. For the shellfish, all the shellfish specimens contained microplastics. Specifically, the whole oyster had the largest amount. The range was from 19 to 78 with a median of about 35. The oyster digestive tract contained much less. It was the median, or the mean, was about 6, and that just is showing you that most of the microplastics in the oyster are likely staying outside of the oyster and not being ingested or incorporated into the tissue. And then the whole scallop also contained microplastics. The mean here was about 16. Thank you.
Thank you.